In this session, we shall detailedly study about the steps of the plant breeding. Let's see the steps of the plant breeding. So this plant breeding is carried out in a systematic way, okay, in governmental institutions and also in commercial companies. They will be doing research. So it is carried out in a systematic way. So in governmental institutions and also in commercial companies. So this will help in coming up with the new cultivars, means new cultivated variety. So those new cultivated varieties that will have superior characters when compared with the old cultivars or with the parents. Anyway, we'll see those things in detail, and by the time we finish of this, we will understand what for purpose this plant breeding will be useful and how we will be able to develop new cultivars. Now, let us look at the steps in plant breeding. The first step is collection of variability. The second step will be evaluation and selection of parents. Then it will be followed by, of course, the third step, and this is cross hybridization. And of course, the cross hybridization among these selected parents, among the selected parents. So cross hybridization among the selected parents will be the third step, and that is again followed by the fourth step. So this is the selection and testing of superior recombinants. So this includes selection and testing of superior recombinants, means the hybrids that we obtain, we need to test them and then select them. And lastly, we'll see about testing, releasing, and commercialization of new cultivars. Okay, so whatever that superior recombinants obtained, we need to test them. Okay, then we need to release them, and we need to commercialize them, right? So anyway, we'll see everything in detail. Now I will be going on discussing each of these steps in detail. So the steps in the plant breeding, the first step is collection of variability, and this is followed by the next one, evaluation and selection of parents. Then the third step, cross hybridization among the selected parents, and then the selection and testing of superior recombinants, and the last one in the plant breeding program is testing, release, and commercialization of new cultivars. The first step in the plant breeding, as I mentioned, is collection of variability. Means variations are differences. Not all the plants will have all the desirable characters. If you look at in the wild plant varieties, you may find one plant that has a property of disease resistance, but it may not have any other okay, desirable characters. Or if you look at some other plant, it has producing a very good quality of okay, grain, but it is not capable of resisting diseases, or is it capable of living in drought conditions. Like this, there are all whatever the characters. So the characters we require, okay, in a plant, obviously the plant should be disease resistance, the plant should be able to give good quantity and quality of the grain, okay, or the product, okay. Then it should be able to resist the drought conditions if possible. If the plant is able to even live in saline soil conditions. So like this, there are a lot of okay, characters, the desirable characters that actually, okay, we want to see in the plants. Now so in the wild we need to check, okay, or in the cultivated crops we need to check whether they have any new characters, the characters that actually we want. We call them desirable characters. And these okay, the collection of variability, which includes the genetic variability, and this genetic variability is the root of any plant breeding. So it means this is the main, okay, point which we need to consider when we're going for plant breeding. Okay, so this is the root of any plant breeding program. So the first step in the plant breeding program, to check the plants with the variations in the genes. So that's what we call genetic variability. Okay, so what that genetic variability, just now I have given you a few examples that the plant is able to live, okay, in drought conditions. Means it has a capacity to live with a very less quantity of water available. So definitely we require such plants, because for irrigation purpose, means the supply of water to the okay, agriculture, I mean to the growing plants in the farm, you can reduce that amount of water. Okay, also like this, there are okay, several requirements of us, and we will be trying to find out any plants with that okay, variations in the genes. Now that's what we see in this case. We will be checking for pre-existing. So the genes are pre-existing, so we'll be seeing or looking for this pre-existing genetic variability. So we are not creating the genetic okay, variability in the plants. We will be first screening the plants that have okay, whether they have the characters that what we are looking for, right? So that's what the pre-existing means, already present in those plants. So pre-existing genetic variability, okay, and that we will see in, try to find out in wild varieties, okay, right, or species. Means they may be different breeds or species, or even okay, the relatives of cultivated crops. So in these plants we will try to find out the pre-existing genetic variability, and this is what we call, I mean this is the actual prerequisite So means to do a program, breeding program, definitely, okay. So this is the step which is very important to be considered, and it is important for effective exploitation of natural genes. So we are not creating any genes, we are only searching for the natural genes present in the plants here, okay, that is available in the, okay, populations. So population, wherever it comes, okay, it is a group of individuals that all belong to same species. So I cannot call, okay, or different species, uh, right, as a population. So here population means it has individuals of, or the members of same species, then only I need to call. So a human population includes only humans, not any other animals, okay, like this, okay. So this is the prerequisite for the effective exploitation of the whatever the genes that are naturally present, okay, in these wild varieties or species or relatives of already okay, cultivating crops and so, right. So this is okay, a small information of the genetic variability, which is a, okay, the root of the breeding programs here. Now the entire collection of plants or seeds having all the diverse alleles for all the genes in a given crop is what we call it a germplasm collection, okay, right. So the entire collection of plant or seeds having all that diverse means, okay, different alleles, which I mean different genes, okay, for all genes in a given crop, what actually we call it a germplasm collection, right. So this is what I call it, the germplasm collection. Statement here again I have mentioned, right. So the entire collection of the plants, okay, or the seeds that have different alleles, I mean that is different genes, okay, right. For all that mean here the genes will have the alternative forms, okay, the alternative forms of a gene, what we usually call it a allele. So the alternative form of a gene is what we call it allele, right. So in a diploid organism, okay, for a gene we will have two alleles. So this little point you may find difficult in understanding because we have not completed the chapter principles of inheritance and variations, okay. So when we come to that chapter, you may get a clear-cut idea what actually those alleles and why I am using gene, or sometimes I'm using, okay, the word synonym with gene with the allele. We'll all see, okay, in the coming sessions. So at present just take this, okay. So the diverse alleles for all the genes in a given crop, okay, we call it germplasm collection. So this is a first step, okay, in the plant breeding program here. The next one is evaluation and selection of parents. So this will be the second step, okay. So whatever the germplasm that we collected, we will be evaluating it for the characters that actually we require. And if we find those plant seeds with desirable characters, then we are going to make them the parents, right? So that's what. Okay, so after that germplasm is collected, I told it will be evaluated, and we will try to identify the plants with that desirable combination of characters. And if we find the desirable combination of characters, then we are going to make them as parents, and later we'll cross them to get the hybrid varieties. That is, the selected plants are now multiplied, means increased in their number, and once we produce them in many numbers, we will be using them in the process of hybridization. We know the hybridization is transfer of the pollen grain from one plant to another plant. So of course that if we do it by ourselves, that becomes something called artificial hybridization, right? So that is, the selected plants are multiplied, which means they are increased in the number, and they will be used in the process of hybridization. And this helps actually, if possible, the development of pure lines here. So that is help in developing pure lines. So pure lines will be created whenever are created, I mean wherever required, and possible, right? So here pure lines are the plants with the homozygous condition. So pure lines are plants with the genes in homozygous condition. Right. Here I'll give you an idea, what actually is this homozygous condition. So if you see, we know in a diploid organism, there will be two alleles, right, for a character. So here you will find, capital A, capital A. If these two alleles are of same type, of course when I mention it's a capital, it may be dominant, and if I mention smalls, it is recessive. Whether dominant or recessive. So this is a dominant, the other one is also dominant, then it becomes a homozygous condition. In the same way, here also the recessive, one is recessive, if other is also recessive, in this case this is also homozygous condition. But if one allele is dominant and if other is recessive, then it becomes a heterozygous condition. So if one is dominant and another one is recessive, that's I'll mention here. So capital A, small a. So the capital A indicates a dominant allele, and this small a indicates a recessive allele. If it is the case, then this is a heterozygous condition. So if the plant has more number of genes in heterozygous condition, then there is a chance of these genes to get separated. So that's what we call it segregation. We do not want the genes to get segregated, they must be present. So if I'm crossing a homozygous plant with another homozygous plant, then I will be able to get again the plant with the same characters in the homozygous condition here. Fine. So anyway, here if you are not understanding also, don't worry about this. Okay, we will detailedly understand when we study about this genetics. Okay, so this is the second step, the evaluation and the selection of parents here. Then the third step is cross hybridization among the selected parents. Cross hybridization among the selected parents, right? So in the second step, we have selected the parents, and whatever that parents that we have selected with the desirable combinations, we are going to now, okay, do a cross hybridization, right? Now let us see, we'll take two bisexual plants here. Okay, so these two are the one that have, okay, the desirable characters. So these are parents with the desirable with desirable characters. So what those desirable characters, I'll just mention the desirable characters here. Say suppose one of these parent has high protein quality, high protein quality, okay, and this another parent, it has, okay, disease resistance, disease resistance, okay. So which means this one is having a high protein quality but no disease resistance, and this is the parent that has disease resistance but no high protein quality. Okay, I don't want to grow these two plants separately. Okay, so in, okay, both the cases I may get, so if I grow these plants, okay, I may be thinking that I will be getting, okay, the quality of the product with the high protein, but as they are not disease resistant, if the disease attack, then the plants will die and the farmer in fact uh, gets a lot loss. Then in this case also the same situation here. So even though it is having disease resistance, I am not going to get this, okay, character. So high protein quality, even though I get a good quantity, the quantity will not be having that high protein quality. So I want these, okay, two characters to be present in only one individual, and I will do it, okay, by cross hybridizing these two parents here. Okay, fine. So that's a cross hybridization. So that is just transferring the pollen, okay, from one flower to present on one parent to the flower of another parent here. So that will actually help in producing the hybrid. Okay, so the hybrid will have these both characters. So high protein quality plus it will have disease resistance. Okay, right. Now the point is that I have given you in just in 1 minute I have told you that just cross the two parents with the desirable characters and you will get a hybrid. Now point is that, okay, after the not just in the first cross, so here I have crossed, okay, then I will be obtaining the seeds, right? And when I sow these seeds, I may have some 1,000 seeds with me, and I have sown all those 1,000 seeds and allow them to grow and come to, okay, flowering, and then see the product here. Now even though I have obtained 1000 seeds, I may not get this hybrid in all those 1000 seeds. Okay, if I don't get, then again I have to, okay, perform this process, okay, and again see the, okay, progeny whether it has this these two characters. If you did not get, again you have to still, okay, you get the hybrid with these desirable characters, you should go on doing the experiment. So for that, okay, problem, this is actually a very time consuming and tedious, okay, process. So that's what I mentioned here. So this step, the cross hybridization among the selected parents, is very time consuming. It requires a lot of time because the seeds you have to, okay, sow them, grow them, and then see whether they have the desirable characters. Okay, so it's a very time consuming and okay, tedious process. Okay, right. So since in this case you have to carry the pollen grain, so you have to transfer the pollen grain from, okay, from the male parent, okay, from the flowers of a male parent, and then to the flowers of a female parent, and okay, you have to follow whatever that artificial hybridization steps which we have seen, like emasculation, bagging, and so, okay, and again rebagging, and then, okay, see the seeds you obtain, and then those seeds you have to sow them and okay, grow them and see whether those plants have this high protein quality and disease resistance. So here, if you are lucky enough, you may get, okay, within right, you know what I call, in the first step itself, or if you're not lucky enough, it may take several times of repeating the same experiment, right? So that's what I say, only one in few hundred to thousand process will show desirable combinations. Okay, so that's what I'll mention here, only, oh, this important point here, okay, only one in few hundred to thousand, okay, process. So here you imagine I'm going to, okay, repeat this process, okay, from few hundred to, okay, thousand times, right? So in a few hundred to thousand process, they show, okay, desirable combination. So that's the problem, okay, with this step, okay, cross hybridization among the selected parents. Right, now let's move on to the fourth step, that is the selection and testing of superior recombinants. Right, the fourth step, selection and testing of superior recombinants. Okay, now this step is a very important step, and of course it's a crucial, okay, step to the success of breeding program, right? So this step is, right, crucial to the success of breeding program. So if we go anywhere wrong, then it can result in, okay, the development of seeds that may not have desirable combinations, and you know, these in the sexual reproduction in flowering plants we have studied, okay, the hybrid, okay, seeds have to be produced every year, of course. So how they are going to produce every year, this is the procedure they are going to follow, okay, every time, and they produce those hybrid varieties. And we know these hybrid variety seeds okay, are very costly, right? And there we have seen, if we are able to make those plants apomictic, then there is no need of okay, going for all this process, and the seeds may become much cheaper. But anyway, okay, so we are that process is still in research. Okay, fine. We'll see this fourth step, okay, which is very crucial to the success of this breeding program here. Now, so in this step, okay, it will yield plants that are actually superior to both of the parents, right? So this step yields, okay, the plants which will be superior to the, okay, both of the parents, plants, okay, superior to both of the parents are obtained. Okay, so because see, we have seen one with the high protein quality and one with the disease resistance. So neither of the parent has both the characters, so but the offspring has both the characters. So for that, definitely the offspring is superior when compared to, okay, both the parents. So here very often this step will yield more than one superior progeny. So very often this step yields more than one superior progeny, which means we get many, okay, superior progeny, okay, with a different combination of characters. So if we feel that those characters are very important, then definitely we will retain those, okay, offspring or the progeny, okay, and we like to develop them further, right? And then see to produce again new cultivars. Whatever that superior progeny is obtained, the superior progeny obtained, now they will be okay, crossing for several generations. In fact, okay, so they will be going for, okay, self crossing for several generations. Okay, so here fine, going for this okay, self pollination. So I'll mention here instead of crossing, okay, self pollination for several generations. So when you are going for this self pollination for several generations, actually it will help in producing, okay, the pure lines. So that's what I said, the pure lines are the one, okay, with the homozygous condition. So this step actually results in yielding, okay, a homozygous condition here. So I mentioned here they perform, okay, this process till they get a state of uniformity, means the uniformity in the genes, a state of, okay, a uniformity, and that's what I call it a homozygosity. So just now, okay, right, I have told you what actually is this homozygous condition. So when all genes are of same type, okay, maybe the two alleles for that, okay, particular right character should be present in homozygous condition. So either it may be dominant or recessive. Then this is the condition what we call it. Okay, so the superior progeny what we obtain is self-pollinated for several generations. Okay, and this will actually help in obtaining pure lines. So that's what here which I mentioned, to get a uniformity, state of uniformity. This is what we call, and simply pure lines. Okay, right. Now after this, the selection and testing of superior recombinants, we will move on to the last step, that will be testing, release, and commercialization of those new cultivars. Okay, so let's see the last step. Okay, so the last step, right? So testing, release, and commercialization of new cultivars. Now in this step, what they are going to do is whatever that superior recombinants obtained, or the superior progeny obtained, okay, so will be evaluated. So they are going to evaluate this all these superior progeny for yield, means how much yield they are able to give. Okay, so of course even at this stage, if anything goes wrong, they have to repeat the whole process again. Okay, fine. Next one, they will also look for disease resistance, and even for quality, etc. So the yield, which indicates the quantity, right? And for disease resistance, quality, like this, they will be seeing whether the desirable characters what they're looking for are present in this superior progeny or not. Now, so for that purpose, they usually grow in research fields. So there will be lot acres of land, and they see that how much yield they are going to get for 1 acre of land, and see whether in the real conditions they have capability to resist the diseases, and what will be the quality of that product or the yield. So all those they will be doing it by growing them in the research fields. And so in this case they are going to even record their performances. So all their performances, they will record them. I mean they will record or keep them in the in a written form, right? And then they do it under ideal fertilizer application. So as I told that they are going to grow in their research fields, and this will be under ideal fertilizer application. So so only that required fertilizer will be applied, and they will be seeing their performance. And also irrigation, so how much water it actually requires, only that much they will be supplying to this growing plants. And also other crop management programs they are going to use. Crop management, practices. I'll write here, etc. Okay. I'll write here practices, etc. will be used here. Okay. So also other crop management practices, so they will be also considered. So under ideal fertilizer application, okay, ideal irrigations and other crop management practices, they will see that how far the plants are able to give their performance. Now once this step is completed, now they are going to grow the plants in actual or real, okay, farmers field. So after, okay, this step, they grow in farmers field. So this is the real time, okay, and see how they are actually their performance in the real time. So earlier they were all under the ideal conditions, okay, they may give, okay, good yield, but under the real time conditions, so whatever the seeds they have produced, okay, they will be growing in the actual, okay, farmers field here, right? And they will be growing in these farmers field for at least 3 growing seasons to ensure that the cultivars are stable, okay. Right, and they are going to grow this for at least okay, 3 growing seasons at several locations in the country. So at different locations they will choose, okay, the I mean soils with maybe with more, okay, salt conditions, or the places where there is less water availability or more availability. So they will be going with the different conditions and see how the plants are able to cope up with the situations, okay, and whether they are going to give the yield, okay, what they actually thought of. All those will be done, okay. So once if they become success, right, now they are going to release for commercialization. So they produce the hybrid seeds that have superior characters, they pack them and then sell them, okay, in the markets to the farmers. So here the farmers will go up and purchase, okay, so and so variety of cotton, okay, of a groundnut type, or there are so many plants, okay, the hybrid varieties are available including tomato, okay, and so, right. So this is how the plant breeding steps, okay, and they are in fact very important for both, right, board, okay, as a 5 marks question usually they like to ask, and also for the NEET